Hello Biology 230 at Tuskegee University. This is Dr. Griffin with video 2 for Module 11. Remember last time we talked about DNA being the genetic material. This was first shown by the Griffin experiment. That DNA in fact is the right stuff, if you will, to program cells and their activities. DNA is simply a polymer, which means it's made up of what? I'll let you answer that question for yourself. But it's a polymer of nucleotides. Adenine, guanine, cytosine, and thymine are the main nucleotides that make up DNA. Nucleotides base pair according to Chargaff's rules. Chargaff, that word is French. I did not take French in high school, so I do apologize for my mispronunciation. The main two rules, let's get back on track. The main two rules are that, number one, base pair composition varies among species. So if there's a question about which organism an organism belongs to, we can actually use gene sequencing to determine that, to make that call. Second of all, the second rule is that within a species, the number of adenine and thymine bases and the number of cytosine and guanine bases are equivalent. Why is it that adenine, or A, must equal thymine, or T, and C must equal G? Well, the basis of this is the classic and famous hydrogen bond. That's right. Wait for it. It's time for a little chemistry blast. Remember, hydrogen bonds are small, weak bonds, but they're very critical in actually keeping molecules together, especially water. But hydrogen bonds actually are the key for holding DNA intact. Now, as you can see, not so clearly in this picture, but adenine and thymine create two bonds with each other, two hydrogen bonds, versus cytosine and guanine make three hydrogen bonds with each other, and thus A and T must always bind together, and C and G are always bound together. If you have anything else, it doesn't make a great bonding pair, and the DNA will not be very stable. Just two main words to describe overall DNA structure is that it is a double helix and it is anti-parallel. Double helix, that description comes from the fact that there's actually two helixes. It's two different strands of DNA wrapped around each other. And you can see this very clearly in figure portion A here. These two strands are wrapping around each other, creating this double helix. The other term, anti-parallel, refers to that the polarity of the two different strands. One strand is going to be running from the 5' to the 3' end. Remember, at the 5' there's the phosphate group, and at the 3' end you're going to have this hydroxyl group that is free, versus the other strand runs from 3' to 5'. And so that is what anti-parallel means, that they're running different directions, but they are indeed parallel in structure, geometrically, with each other. And this is, you see in the chemical structure and the space filling model, the double helix, especially represented in the space filling model, as well as the anti-parallel nature of DNA. And you see very clearly here the two hydrogen bonds made by adenine and thymine, and the three hydrogen bonds made with cytosine. Wait for my mouse to come back up, my cursor. Cytosine and guanine. Okay, here's a question. Which pairing is going to be stronger? Adenine and thymine or cytosine and guanine? Think about that. Well, that question ends video 2 out of 4, so you're almost there, halfway there for finishing the videos associated with Module 11. This is Dr. Griffin signing out, and I look forward to helping you learn from the next video.